mRNA molecules are a carrier of genetic information that cells use to produce specific types of proteins. This property determines the functioning of cellular processes, playing a key role in the life of organisms. Modern medicine pays a lot of attention to mRNA because it can form the basis for creating new, advanced gene therapies, including personalized ones. mRNA can also be the foundation for designing innovative vaccines. However, researchers face a challenge in the form of the instability of mRNA and the limitations in monitoring the behavior of these molecules in cells and organisms. This is why scientists from the University of Warsaw have developed a universal method of fluorescent labeling of mRNA. It allows the precise verification of what's happening with these molecules once they are in the organism. The structure of mRNA resembles a thread. Its main part is the nucleotide sequence. Due to the high differentiation, these molecules take longer or shorter forms, but they always have the same endings. One of the ends is called the 3-poly A tail, and the other is called the 5-poly A tail. The invention from University of Warsaw allows to attach to both of these ends fluorescent tags. The developed method is universal, which means that it can be applied to any modified mRNA molecules consisting of any number of nucleotides coding for any protein.
Opracowana przez nas metoda znakowania fluorescencyjnego mRNA.
Our method allows the universal fluorescent labeling of both ends of the mRNA molecule at the same time. As long as the labeled mRNA chain is uninterrupted, both dyes at its ends communicate with each other, and the images from both dyes overlap. However, if the molecule is broken, for example as a result of the natural action of a degrading enzyme, one part of the broken molecule glows blue, and the other glows red. In addition, if the mRNA encodes green fluorescent protein GFP, we can simultaneously observe the formation and distribution of the synthesized protein and the state of the mRNA. The innovative labeling method allows for direct and rapid one-pot modification with high efficiency from 65 to 99%, depending on the length of the labeled mRNA molecules.
Takie narzędzia są niezwykle przydatne dla badaczy.
Thanks to this, in laboratory conditions, it's possible to determine very precisely how mRNA behaves in living cells, and even in some organisms. Researchers with such effective monitoring at their disposal can make the right decisions much faster in terms of further research and development of technologies used in the designed therapies. The developed method is easy, fast, highly effective, and most importantly, inexpensive, and it doesn't require the use of enzymes or catalysts. To date, no alternative method has been developed in the world that would allow for fluorescent labeling of mRNA in such an effective manner and with such high efficiency. The developed method is ready for use in scientific research, biotechnology industries, the pharmaceutical. It's made available based on licensing agreements. The presented technology is protected by law. Contact us to discuss the details.